Next topic is the neuroglial cells. Apart from the neurons, the nervous tissue consists of vast supporting cells known as neuroglia or glial cells. They play a supportive role in maintaining the nervous tissue stroma by filling up the space between the neurons. These cells completely envelop the nerve cells and provide functional as well as structural assistance to the nerve cells. They also provide insulation to their neurons and their processes, but do not contribute in the propagation of impulses or processing of the information. Unlike the nerve cells, the neuroglial cells are capable of multiplying and fill the gap between the neurons. So brain tumors taking origin from these cells are termed as gliomas. So the neuroglia in the CNS are classified are as follows: ependymal cells, astrocytes, oligodendrocytes, microglia. And the neuroglia of the PNS, Schwann cells and satellite cells. So as the neuroglia are derived from the ectoderm except the microglia, which are derived from the mesodermal in origin. Now we will talk about the neuroglia of the CNS. First is the astrocytes. They are the largest and most numerous cells of all, and they form the main supporting cells, and because of the shape they are called as astrocytes, and they are star shaped. Structurally, they have long dendrite-like processes with small swellings at the end of the food processes. And astrocytes are involved in the exchange of metabolites between the neurons and capillaries, and they are considered to be the storehouse of glycogen in the brain. Embryologically, if you see, they are derived from the neural crust, and there are two types of astrocytes: protoplasmic astrocytes and the fibrous astrocytes. So what are protoplasmic astrocytes? These type of cells mainly found in the gray matter of the central nervous system, and they have thick processes with abundant granular cytoplasm. And in contrast with the fibrous astrocytes, which are mainly found in the white matter, and they have long slender straight process, and they are less branched than the protoplasmic astrocytes. And these fibrous astrocytes take part in the formation of blood-brain barrier. And these astrocytes are absent in the pineal gland as well as the posterior pituitary gland. What are the functions of these cells? They provide mechanical support to the neurons. They provide nutrition to the neurons and play important role in the transportation of the metabolites and waste products to and from the brain, as they are storage places of glycogen. At the region near the blood capillaries, the food processes of the fibrous astrocytes form perivascular feet around the capillaries. Hence, the blood gets separated from the neurons via endothelial layer of the capillary and the layer of the astrocyte food processes, together forming the blood-brain barrier. They form tight junctions in this area with the endothelial cells of the capillaries. They aid in maintaining the environment which is favorable for the impulses to generate, but they do not participate in the impulse generation and prevent the spread of nerve impulse in unwanted directions. They also undergo a process called as gliosis, that is, they undergo hyperplasia and hypertrophy when the nerve is injured and fills up the void area to form the hardened scar tissue. They promote the growth of the nerve by secreting the protein, nerve growth factor, and also metabolize excess neurotransmitters. And next are the ependymal cells. The ependymal cells line the ventricles of the brain and also the central canal of the spinal cord. These cells are arranged in a single layer fashion, and they do not form tight junctions, and hence the molecules can freely pass between the CSF and the nervous tissue. Embryologically, they are derived from the neural tube, and there are totally three types of ependymal cells. The first one is ependymocytes, second is the choroid epithelial cells, and the third one is tanycytes. The first one is ependymocytes, or cuboidal or columnar cells with the cilia and microvilli on the apical area of the luminal surface, and they constitute the main form of ependymal cells. And the second one or the choroid epithelial cells. These are specialized type of cells, or we can say specialized form of ependymal cells where they secrete the cerebrospinal fluid. And next is tanycytes. These are the cells lining the floor of the fourth ventricle with long basal processes. And what are the functions of these ependymal cells? Ependymal cells are responsible for the formation of the cerebrospinal fluid and assist in circulation. They also take part in the transportation of dissolved gases and nutrients and other materials between the brain tissue and the CSF. And next is oligodendrocytes. Oligodendrocytes are small, round and oval cells with few cytoplasmic processes. They are present only in the central nervous system and are found around the neurons of the grey matter and along the length of the myelinated nerve in the white matter. Embryologically, the oligodendrocytes are formed from the neural tube. And what are the functions? Oligodendrocytes form the myelin sheath around the axons of the central nervous system, whereas Schwann cells forms the myelin sheath in the PNS. So a single oligodendrocyte forms myelin to the multiple neurons of the central nervous system. They also form the supporting tissue around the nervous tissue. And next is microglia. Microglial cells are the smallest of the glial cells. That's the reason the name is micro. And they are involved in the phagocytic activities of the CNS. They are generally few in number and migrate in the neural tissue when there is any ongoing infection or inflammatory process. Embryologically, the microglia is derived from the mesoderm, similar to the origin of the monocytes and macrophages of the blood, and migrates into nervous system before birth. And what are the functions of these microglial cells? Microglia enlarges during phagocytic activity and engulf the invading pathogen and clear the debris during inflammation. They can retract and migrate to the areas of the degenerated or damaged nerve tissue and are highly active in degenerative diseases like Parkinson's, Alzheimer's and multiple sclerosis. And if you see the neuroglia of the peripheral nervous system, they are satellite cells, Schwann cells. So what are the satellite cells? The satellite cells surround the nerve cell bodies of the peripheral ganglia, that is the spinal and autonomic ganglia. They are flattened cells with prominent nuclei and it gives support and nutrition to the PNS. They are present in the peripheral nervous system only. And next is the Schwann cells. These are also called as neurolemmocytes. They form myelin sheath around the axons in the peripheral nervous system, and these are flattened cells with a flat nucleus and abundant cytoplasm. Embryologically, if you see, these are derived from the neural crust and found only in the PNS. What are the functions of these cells? Schwann cells produce neurolemma around all axons in the peripheral nervous system, both for the myelinated as well as non-myelinated axons. They also help in regeneration of the axons in the peripheral nerves. This is what we need to know about the neuroglial cells and its functions.